All right, Fred, you're up next. Help us understand the science a little bit more deeply and perhaps some of the controversial viewpoints here.
Okay. So, thank you all for sticking around. This is amazing. So, Friday afternoon, my initial intention was to really explain some of the nuts and bolts of different gene drives to you on Friday afternoon. But with all of this happening yesterday, I changed my talk at around 4 o'clock this morning to focus a little bit more on that and give you just a quick overview. But just to say, what I wanted to talk about is gene drives from species eradication, where you might want to eradicate a mosquito, to species preservation, where you might want to preserve endangered species like these Hawaiian honey creepers, and we can get into that. But I also just want to say that I'm from the Genetic Engineering and Society Center at North Carolina State University, and our sort of tag is that we integrate scientific knowledge and public values. And we can discuss that as we move on. But what I want to do is go on and just tell you a little bit about gene drives and explain some of the issues, but just very generally. All right, so people have been able to genetically engineer mosquitoes so that they can't transmit dengue virus. But the recognition is that if you have a big population of mosquitoes and you release your happy laboratory mosquito here, it's not going to do much to decrease dengue fever. So what you want to do is you want to hook that gene up for not transmitting the disease to something that has what's called super Mendelian inheritance. Instead of 50% of the individuals carrying that gene to the next generation, close to 100% can with some of these new technologies. So there are a lot of different ways of doing this, and one is called unrestricted gene drive. So you release this one, and it will first transmit into local individuals, but some of them will move to a population nearby and will also spread. Okay? And the concern about this since 2003 has been a real concern for researchers, is that if these things start spreading like that, they'll spread throughout the range of that insect. So they might make all of the Anopheles mosquitoes not transmit the disease. But you could also rig this thing so what it's doing is it's slowing down reproduction, and that could lead to extinctions of a species you don't want. So the question is, you might not want it, but maybe other people do, and are you playing God here? So that's one approach to gene drive. But another is what's called spatially restricted gene drive, and there's another mechanism to do this, and you can use CRISPR or other things where you release this one, and it spreads in that local population, but that's as far as it goes. So if you wanted to get rid of mice on an island, you use something that's specific, a gene drive that's specific for those mice, but doesn't work on the mainland. All right, so it stops there. Another approach is called threshold restricted gene drive, where you can't just release one individual, you have to release a large number of individuals, so you have something like 20 to 50% of the population having that drive, and then it can move in, otherwise it doesn't. And that's considered to be a very safe kind of gene drive because it is so restricted. So you can release something like that and it will spread, but again, you're not going to get that large threshold in the next population if only a few move into that population over time. So that's the basic rundown on gene drives. But what I was talking about before was, let's see if I can go back, was mostly using this to transmit something so the mosquito doesn't transmit the disease. But I also mentioned that you could use this to suppress a population. And one of the ways that people have talked about suppressing a population is having a trait where the females don't produce eggs. That's a pretty simple one. Another one, though, is to release a male mosquito here that has specific genes that change only cause the female to produce more males. So you start off with a population that has males and females. You wind up with a population that has only males, and we know what happens when you only have males, right? So the idea is that you would have suppression of the population. That's a very different approach in terms of the population genetics than putting in something that just changes the characteristics of the population. Okay. So, moving on to yesterday. Yesterday, there were 2 articles released. This one was under embargo, up until 2 o'clock yesterday afternoon called Conservation Demands Safe Gene Drives by Kevin and a colleague. And this is published in PLOS Biology as an opinion piece. And then another one backing this up was published in bioarchives, meaning it was not yet peer-reviewed. Okay, so there are those two papers, and taken together, those led to this yesterday. Genetic this is the news coverage, and this is what we want to talk about: is how do you go from research to news coverage, and what can we learn from this case study? So all of a sudden, you went from those two papers to this in Gizmodo, which I don't read. I don't know how many people do, but this is in the New York Times by Carl Zimmer. Right? This is in the Atlantic. And that New Zealand wars on rats could change the world, okay? So look at what's happened and look at the pictures. Somebody brought up images that are used. And this is probably the most balanced one in Quanta magazine, but look at that image. I'm not sure exactly what people falling off DNA strands has to do with that, but you know, not the same person writes the article as puts that out there, or the title. So what are these? What's the deal here? What was in these? So I just extracted this from the abstracts, and the first paper says New Zealand now considering gene drives as a way to locally eliminate mammalian pests. As you saw those pictures in the articles. This article, right, will explore the risk of accidental spread following deployment. Need open and international discussions concerning the technology that could have global ramifications. Now, the research community responded differently than the magazines, because a lot of this stuff, the people in New Zealand say, whoa, we're just looking at all kinds of approaches. But New Zealand, if you think about GMOs in New Zealand, they don't go together. So they're just thinking about this. They're not really getting on board. They just wanted to not throw out anything. And there can be accidental spread. The international discussions, of course, have been going on since, I think, 2008. So this was a little bit, well, where was this coming from? And then the other report said that even the least effective system reported to date are highly invasive. And we'll get to that. And then, contrary to the National Academy's report on gene drive, our results suggest that standard drive systems should not be developed nor field tested in regions harboring the host organism. And remember, this is not a peer-reviewed publication yet. It's a mathematical model. So this is the best gene drive that's been tested so far. And in 2015, this group presented this data in Nature Biotechnology, a high-end, high-impact journal, showing an increase due to the gene drive. But then they recently published the fact that this thing fell apart in the laboratory. So not quite ready to release in the field, and not very effective at invading the laboratory. So I don't know about invading the field. How am I doing on time? Okay. So the other thing that the comment was that, contrary to what the National Academy said, this is the National Academy Gene Drives on the Horizon report. The subtitle, Advancing Science, Navigating Uncertainty, and Aligning Research with Public Values. So if you actually read this report, you'd see that they cover a lot of this stuff in a lot of depth. So I said this was the most balanced one. This is the only one of the reports in the, among all those reports I showed you, that actually asks a question about the mathematical model. And it says, but the models don't consider the gene drives that most scientists think are more likely to be used in conservation projects. That's called suppression drives. In those, the gene drive alters a trait that the population needs to survive. For example, such as a drive that may skew a population to be all male or all female, it stops breeding and dies out. It's unclear whether suppressive drives would prove as invasive as what was in the model, although Noble, the first author, said he plans to investigate these soon. So not only do we have something that hasn't been peer-reviewed, it's actually not about the conservation system. So I guess sort of the question is, how does these two papers come together and become such a big news story? And the other piece is, you know, where are the responsibilities of the researchers and the journalists in this? So one of the things, this is very controversial, as you heard, in terms of the responses of people to this. And this is what's happening now in NGO communities. This was an article put out, Reckless Driving, Gene Drives, and the End of Nature. So there's a lot going on here with the Cartagena Protocol and other things where these kind of technologies, even for malaria or anything like that, are being pushed to the brink in some ways. So the question is, in my mind, you know, where are, what are these responsibilities? How does something like this happen? And we've seen this happen before. It's not anyone's fault necessarily, but we haven't figured out how to follow some kind of rules of the road, and I hope we'll have a discussion about that. So I'll end and thank you and just tell you that, you know, that's what we try to do in this Genetic Engineering Society Center, is try to improve the way we engage with the public and the way we report scientific findings. Thank you.
Thank you, Fred.